All right, it's my pleasure to introduce our first presenter today, Dr. Ashley Powell. She received a Bachelor of Science in Marine Biology and Chemistry from Roger Williams University, and then her Doctor of Veterinary Medicine from Atlantic Veterinary College. She is currently an aquatic and anatomic pathology resident at the University of Georgia under Dr. Al Camus. So thanks, Ashley, for being here, and looking forward to your talk.
Thank you. Thank you for the introduction. Hello, everyone. Good afternoon. I'm super excited to be here and talk to you about immunohistochemistry in fish health investigations. And so that's my screen. Come on, you can do it. Briefly, immunohistochemistry is the use of an antibody to identify a specific antigen within a target tissue. So there needs to be some idea of what to look for. IHC uses paraffin-embedded formalin-fixed tissues as we would for histology. It's relatively quick and cheap, widely offered by diagnostic labs, both human and animal, and doesn't require fancy equipment, although that being said, it can be automated or done by hand. The most commonly used antibodies are IgG or IgM, and they're developed from either injecting a host animal with an antigen, that host animal mounts an immune response, and those antibodies that the host produce are harvested from the host, and that's what we use. Alternatively, DNA sequence can be sent out for a protein, and an antibody could be synthetically manufactured. There's two types. Monoclonal antibodies are made from a single antibody, usually B cells from a mouse spleen, and it's an immortal cell that produces immunoglobulin for a single antibody isoform or epitope. These are expensive, but have a higher specificity, and it reduces cross-reactivity with antigens. Polyclonal antibodies consist of a mixed bag of antibodies. They're cheaper, more likely to identify different epitopes, so they've increased cross-reactivity. However, there's an increased chance of false positives using polyclonal. Their IHCs have various uses in domestic animals. Perhaps most commonly in the diagnostic setting, they're used for cell origin or identifying specific cell type. For example, they could be used to differentiate epithelial versus mesenchymal or connective tissue cell origin, or more specifically, they could be used to differentiate B cell or T cell lymphocytes. They're used to identify specific pathogens or pathogen families. For example, chlamydia will cross-react against different chlamydia species in different animals, whereas some are more species specific. Not so much in fish, but in humans and domestic animals, maybe someday in fish. They are used as a prognostic indicator. So Ki67 is a predictor of cell replication, indicator of cell replication. So it could be determined how a tumor behaves or predict the response to treatment. We can also evaluate an antibody in multiple tissues on one slide. And this is a multiplex assay, which cuts down costs and allows for a lot of samples to be run at once. Or we could evaluate a lot of antibodies on one piece of tissue using an MI chip. And once again, not routinely used in fish diagnostics. These are the IHCs offered at UGA. Other labs have more, other labs may have less or different antibodies. But these are the ones that we offer here at UGA, and it illustrates the potential out there for fish health investigations. Commercially available antibodies are validated in one or multiple domestic animal species, and they're tested for specificity, how selective they are, and how reproducible they are using different buffers and different methods. The most common components of antibody validation is the use of Western blot, blocking peptides, negative control tissues, and negative control tissues are preferentially from knockout animals. And those are done for more of the routinely used ones, not so much in fish, but I'll talk about that later. And information for those validated antibodies should come with the antibody specificity, the dilution, antibody retrieval method that's recommended. We'll get to that a little later on. Detection systems, interpretation of results, and hopefully some references backing up that information. So you have a fish, and then the fish dies. Then you collect the tissues in formalin, let them fix overnight, or at least 24 hours. And then those tissues will get embedded within a paraffin embedded block. So they're embedded within paraffin wax, and that is what we'll use for immunohistochemistry. But that's what we'll use for routine histology. And this would be an H&E or hematoxin eosin, like a routine stain, what we use for diagnostics. This is going to be a quick overview of what we're about to go over. There's no time to go over all the methods, all the reagents, but just know that there's quite a bit of variation. If you've done IHTs, this might be different than what you're used to. If you haven't done IHTs, the methods are comparable to ELISA's. And if you haven't done both, I got nothing for you. There are two methods. There's direct and indirect. So the direct method, I'll point out, stops after the addition of one antibody. It's simple, but lacks a lot of specificity, rather lacks sensitivity for detection of most antigens in a diagnostic setting. And then the indirect method is most commonly done by diagnostic labs. It's a little bit more complicated. It uses two antibodies. The primary one is not labeled, allowing it to retain its activity with the antigen, and it allows amplification of that label. And so first, an antigen is retrieved from the formalin-fixed tissues. This is done by exposure to heat, enzymes, or buffers to undo the cross-linking caused by formalin. And then all potential binding sites from the tissue have to be blocked to prevent nonspecific antibody binding. And so blockers are buffers that sometimes contain proteins and compete with the antibody for binding to nonspecific sites. The primary antibody is then introduced, binding the target antigen. And the direct method would stop here. And then you would add your substrate, chromogen, enzyme to create a detectable reaction. I'm going to continue with the indirect method, but just know that the direct method would stop here. And then a second antibody is linked to the first antibody. And it's often created in a different animal. So you'll hear things like rabbit anti-antigen and then goat anti-rabbit, and that's what that means. For this next part, and this is the key part for both processes, an enzyme, a substrate, and a chromogen produces a colored precipitate, which is what we're going to view on the slide. And so there's many methods, many substrates, many enzymes, and many chromogens that can be used. And that protocols can vary between labs and vary between antibodies. And there's different advantages and disadvantages to each one. So your second antibody is then biotinylated, or comes biotinylated, and then a substrate and enzyme are introduced. The most commonly used enzymes are horseradish peroxidase, alkaline phosphatase, and the common substrate options are avidin biotin, streptavidin biotin, or peroxidase antiperoxidase. I use streptavidin here because that's what we use for cytokeratin, which I'll talk about more later. And then that binds, a chromogen is then added. And so that chromogen one's commons are DAB or AEC. If you're doing a fluorescent protocol, then you would use a fluorophore. And then that chromogen reacts with an enzyme to produce a colored insoluble precipitate, which is what you see. So there are three common labeling patterns. There is nuclear, cytoplasmic, and membranous. And so they're pretty much self-explanatory. Nuclear, all the little nuclei are highlighting. So these were probably not the ones that were targeted, and that's why they're not marked. But these are. And then here, the cytoplasm is marked, but the nuclei, those little blue ovals and circles, they're not marked. How a reaction should manifest should be provided with the antibody, although it can be predicted. If the thing you're looking for is in the cytoplasm, you can expect the cytoplasm to stain. Immunohistochemistries are mostly described in zebrafish and koi. This is not an exhaustive list. Some fish-specific antibodies are available, and they are somewhat diagnostically validated, such as BKD, PKT, PC rickettsia, and KHV. There's also publications for others. Pan-cytokeratin is the most commonly, I would say, that we use, and it labels epithelium, as in the skin, gills, gastrointestinal tract, or epithelial-derived tumors, so carcinomas. Pan-cytokeratin means that there are a bunch of keratin types within the mix. There are many different types of keratins, and this just contains a bunch of them. S100 is a non-specific marker for Schwann cells. It's used to stain neural tissues in zebrafish. It'll also mark other cell types like macrophages or melanocytes, so it's not overly specific. And various other immunohistochemical markers have been used with varying success in fish, and these include markers for germ cell tumors, endocrine tumors, muscle, and so on. Just a few examples. Melon A stains neural crest cells and can be used to identify pigment cell tumors from other similar looking cell types. So it could identify melanophores, erythrophores, xanthophores, but not differentiate them. And so the chromogen here is red, and that's why the cells are red as opposed to the brown ones we saw earlier. And if you attended Eastern Fish Health Workshop, I presented this case. We identified proliferative skin lesions in a sand tiger shark in a public aquarium. We weren't sure if the proliferations were from the epithelial component or mesenchymal component of the dermal denticle. And so that's when immunohistochemistry came in. So these are those proliferations, and these are the cells, and we wanted to see what cell origin all these cells were. So we have positive cytoplasmic immunoreactivity for cytokeratin, identifying these cells as epithelial in origin and identifying them as possible ameloblast-like cells that produce those dermal denticles. This is a control in a normal developing dermal denticle. And so the epithelial component is highlight, but the mesenchymal component is not. And so you would expect that this was a mesenchymal cell tumor, this portion would be the ones that would be highlighted with a different immunohistochemical marker, such as vimentin, but we'll talk about that later too. Immunohistochemistry, again, using cytokeratin, can be used to show that Hennegaia exilis. So all over here, those are your little myxospores, exist within the gill epithelium rather than outside of it. So this is more highlighting the exact location of that myxozoan. Immunohistochemistry can also be used to detect hard to see organisms. This is a case of Francisella within a damselfish. Normally the bacteria are very difficult to visualize, but highlighted with a Francisella marker, you could see the positive immunoreactivity over on the image to the right. Viruses as well. And so this is a papilloma from a sea lion, and this is a papilloma virus immunohistochemical marker. And this papilloma virus is general across species, so just marks papilloma viruses, not specific for the sea lion papilloma virus. IHC is a tedious protocol that is time and temperature dependent. Errors can occur at pretty much any step. Insufficient antigen retrieval, inadequate blocking, pH of the buffer, type of antibody, time of contact can all cause no staining, non-specific staining, or aberrant staining, or rather marking would be the appropriate term. Positive and negative controls are needed. We use a mammal and a fish one for every run. And using tissues with more than one tissue type within it allows for in situ positive and negative controls. And that could really help for evaluate for specific marking. And then of course, cover slipping, the very dreaded phrase, cover slipping. Limitations in fish, and there are quite a few significant ones. First, there's a lack of commercially produced antibodies that are validated in fish. And a lot of those antibodies are produced in mammals. So as you can imagine, they're not going to necessarily have the same cross-reactivity as if they were developed in fish. There's lack of published literature describing the staining characteristics of fish tissues, how fish tissues are supposed to stain. And given the large variation of fish species, antibodies that appropriately react in one species may not work in another, and therefore individual species may require their own unique set of protocols. Selection of monoclonal versus polyclonal antibody can also play a role in detection. This is routine staining for epitheliocystis. This dude here. And so on the left is a monoclonal antibody that did not work, and on the right is a polyclonal antibody that did. So likely from the increased chance of cross-reactivity within that mixed bag of antibodies for chlamydia detected this in this case. And sometimes everything reacts. This is aberrant staining. The gut epithelium is marked. The blood vessel walls are marked. The connective tissue is marked. The smooth muscle down here in the gut wall is marked. And this is a marker for endothelium. So the cells that line blood vessel walls. So technically only these lining cells should be marked, and they're not. So this did not work. On the left is a dog tongue. So this is the top of the tongue where all those little papillae would be. And this is the muscle that moves the tongue. And so the striated muscle within the tongue is marked, but nothing else. And so that is appropriate specific staining. On the right is gut from a fish. And yeah, while the muscle in the gut is marked, there's also marking of the epithelium, the blood vessels. And so there is non-specific staining, and this calls into question the overall specificity for this antibody in fish. And it really shows where antibody validation and description of expected staining is needed. So this may be okay, or it may not be, and further research is needed to look into that. Antibody dilutions or prolonged incubation overnight in buffer may be needed in fish, and this might be used to prevent non-staining that you saw in the last image. Also note that diagnostic labs may use automatic stainers, which might be inappropriate for these extended protocols. They may need to be stained by hand and let sit overnight to allow that sufficient blocking. And also pH modifications might be needed in fish versus mammals. Why should we bother us developing IHCs in fish? Well, fish are really used in carcinogenesis research. And so immunohistochemistry is useful in differentiating tumor types. It can also be used for pathogen identification, as we saw previously, and could be used to identify poorly differentiated or complex tumors, which is what we're working on now. And so we're working with our histo lab to develop a few more functional immunohistochemical markers. Ideally, we'd like to develop them for both teleost and elasmobranchs. We're trying to target markers that could be used to differentiate these different cell types. Smooth muscle actins, well, for smooth muscle. Desmin is general muscle marker, and vimentin is a connective tissue or mesenchymal marker. And we would like to describe these protocol modifications that can be used, and hopefully classify previous cases. So spindle cell tumors, and the reason why they're such a headache is they're all mesenchymal in origin. And going down the list, so you have those that originate from fibroblasts, blood vessel walls, blood vessel linings, blood vessel pericytes, blood vessel linings, and nerves. And they all look the same. In humans and domestic animals, immunohistochemistry and ultrastructural techniques like electron microscopy are gold standard to differentiate them, and we don't have that luxury in fish. This is from an Alchemus publication and demonstrates poorly differentiated tumor in an Arabian carpet shark. So these are your tumor cells here. And the smooth muscle actin shows positive cytoplasmic immunoreactivity, indicating that this tumor is muscle in origin. Haikui is a spindle cell tumor described in koi. Publications note that these tumors are also smooth actin positive, but we have been unable to replicate these results. So this is one big zero. So stay tuned. We're still trying with those modifications. And we have experienced / are experiencing multiple cases of spindle cell tumors and spotted eagle rays. There's been no luck with muscle, connective tissue, smooth muscle actin, or blood vessel endothelium stains. Even though this is staining, it's non-specific staining was in the slide, so this didn't work. And so we can't yet demonstrate the origin. We think it's fibroblastic. It does have resemblance to scar tissue, and it looks that way with electron microscopy. But for right now, elasmobranchs are a lost cause. So more on that in upcoming studies. So I'd like to thank Alchemus, Elizabeth Howarth, who is our IHC queen, and the UGA Histology Lab for their patience in this endeavor. And here are my references.
Awesome. Thank you very much, Ashley.
Thank you.
Folks, if you have questions, feel free to drop them in the chat or just unmute yourself. You can ask her directly. I'll start with one, Ashley. What's the turnaround time and expected costs, like typically from a diagnostic lab for performing an IHC?
Yes, that's a great question. So turnaround time, we run them daily. So sometimes there's delays if they're doing quality control, but normally it takes about 24 hours to get the slides, from the request. So they'll run it that same day if you get the request in by 9 a.m. So, it's rather quick. And then cost, our diagnostic lab for our domestic cases usually charges between $60 to $70 per slide, but they also offer like a bundle. So it could be like $112 for 2 stains. Mm-hmm. So it's relatively cheap.
Yeah. And for these complicated cases, I could see where that'd be really valuable.
That's a good question.
Hi, Ashley. This is Jeff Wolf.
Hi.
How are you?
Good, you?
Great. That was a wonderful talk. I really enjoyed it.
Oh, thank you.
Do you guys use isotype reagent controls, or do you omit the primary antibody? And do you want to talk about the difference between the two?
We have not done that. That is a question for my lab. Have you had success in doing that? I believe our lab will run. I really am not familiar. I'm sorry.
Well, the isotype control is another guard against non-specific staining, where you actually, instead of you use the same type of antibody like IgG or IgM, but it's not specific for anything. And that instead of omitting the primary antibody, that allows you to, make sure that you don't have non-specific staining by the group of antibody that you're using. And what we've seen is that when you do that, all of a sudden there's a lot of staining that you thought was specific that probably really wasn't. So
Gotcha.
that's kind of been our experience.
You know, that's an excellent point. I'm going to investigate that with our histology lab. Thank you.
Actually, you have a question here in the chat from Bart. He's curious about the most common IHC requests that you get for diagnostic cases.
So we don't get many. And so for requests, not so much. We're doing this mostly for research purposes and things we can offer. Often we're the ones to, you know, we will get a mass submitted to us. And we're often the ones asking the question of what is it? We run cytokeratin most often because it works fairly consistently. We have also used chromogranin A. We've used that for like endocrine tumors in koi. We've tried a variety of neural markers, so like neuron-specific inalase. We've used ones for myelin, but we've had GFAP, but we've had varied success with them. So this is something that's really new and something we're really trying to develop so we can offer it and we can answer the questions as to what these things are. But right now it's super limited. And yeah, unfortunately, that's the best I can answer that question.
That sounds good. Any other questions out there? All right, great talk, Ashley.
Thank
you
you.
very much for presenting.